Alrighty, hi everybody. Um, Thank you for coming to the talk. I really appreciate it. I'm going to tell you a story that I haven't spoken about yet. So it's a first time talk, and I always find those a little tricky, but we'll see how I stumble through it. Um, First and foremost, though, I wanted to thank BioLegend for inviting me to do this. It's really a fantastic opportunity for somebody so junior to be able to, talk to people and talk about their work. When I was talking to BioLegend about what to do for this talk, they said, okay, well, what are you doing in the lab? And I said, well, here's the stories that I have in the lab. And they picked the only story that's very conventional flow cytometry, which is fine. But I don't think that that actually showcases the great things that BioLegend has done for us. And they said that I don't have to plug them, so I'm not being held at gunpoint or anything. I'm choosing to do this. I'm on the steering committee for the IBEX imaging community, and we do high-plex, multiplexed imaging. And you're not going to see very much of this in this talk because, again, this is a very flow cytometry conventional talk. But we're able to get like really high-plex images. The incredible student who's done a lot of this work is sitting right there. Her name's Victoria. She's amazing. She has a poster out there. And BioLegend's been fantastic with the imaging community, from helping with reagents to even creating repositories on their website for it. So check them out and check those things out. We also do a lot of like site-seq work and use those reagents too. And so, we interact with BioLegend a lot, but this is a very conventional flow cytometry story. So I study IgE antibodies. And anybody who's taken a class in immunology, which I assume is most people here, has seen something like this before. We have these IgE antibodies. They bind very tightly to the surface of mast cells and basophils, and when they see their antigen, they immediately degranulate. They release pro-inflammatory mediators that cause the signs and symptoms of an allergic reaction. And so it's not super surprising that we see this as the target in this disease. We want to try and eliminate IgE production in these antibodies. And in doing so, you would eliminate the degranulation that happens, and ultimately you would be able to eliminate the disease. Easier said than done. And I think one of the reasons for that is because targeting IgE is not trivial. So omalizumab, which is an anti-IgE antibody, was just approved for food allergy. And oral immunotherapy, taking an antigen, it can reduce IgE. But the problem is, if you stop those therapies, those things come back. And so the way that my lab is thinking about things right now is that we need to target not just the IgE antibodies, but the reservoirs that those IgE antibodies come from. So there are plasma cell reservoirs and IgE plasma cells that secrete these IgE antibodies. Most of the literature around these would suggest they're pretty short-lived, but there is some evidence that suggests they can be long-lived. So I don't want to only show one side of the coin there, but I'll say that from in our hands anyway, they tend to be pretty short-lived. But what's pretty clear is that memory B cells, when they experience their antigen, they can become IgE plasma cells and replenish that repertoire. And so for us, that's why we're really focused on looking at this memory response. We need to understand how the immune system knows to create more IgE constantly. Now, if you study vaccination, the type of work that I want to do is not too hard. You can get people, you can get their pre-immune repertoire, you can then immunize them, and you can bleed them and see, for example, how memory is initiated, right? You can see how the memory B cell response starts, for example. I think our ethics board would have some concerns if we tried to make people allergic to do the same thing. So we need to do natural experiments in humans to do some of this work. And the problem is that people show up to the clinic already allergic. We don't have a way to capture people before they're actually allergic to see the progress of that memory starting, right? So they're already anaphylactic. And the other really weird thing is that most often it's on the first known exposure to the food. That's what the parents will say, that the kids never had the food before, but we gave them the food and then they had anaphylaxis. And so trying to understand the induction of memory, how we get these memory cells, it's really hard because of this. And so some people have tried, they get big cohorts, they find who becomes allergic, and they took a look at samples over time. And this is one study where they took a look at the emergence of IgG antibodies by comparison to IgE antibodies. And you see that the IgG comes earlier. This here on the y-axis, it might be hard to see, things might be small, but this here on the y-axis is the prevalence of these antibodies. And you can see that they start, a few years before the emergence of the IgE happens. So the process of sensitization is probably not one event that happens really quickly. It's probably a phasic thing where things occur over time and develop as the immune system develops. And we were able to replicate some of this trajectory in mice. So when I was a PhD student with Manel Jordana, I contributed to this work here, where we used a tape stripping model and we could induce IgG. And then you needed to re-expose the mice further after the tape stripping to start an IgE response. So there is a predating of IgG immunity to IgE immunity. But we are still not really able to see how this allergic memory starts, how it's seeded in this process. And so what we really wanted to do, and what the beginning of this whole project was, is we wanted to understand basically these beginning steps of seeding these memory B cells. And you know, if there are any B cell biologists who are here today, most people will say, well, that's fine, just immunize a mouse and then take a look at the primary immune response to see the generation of memory. And that's a little bit more difficult with these allergen sensitization models because they don't tend to just take one exposure. So what is a primary exposure in this system? If you need 4 gavages to make a mouse fully allergic, that's not the same as like a single gavage exposure. So what is the kinetic of that response? And what it told us is we needed to start looking within these models and understanding the trajectory of these models. So here's what you would see if you had an allergic mouse with these 4 OVA-CT. Some people will do up to 6 OVA-CT gavages. You know, the mice will make OVA-specific IgE and IgG1, and if we give them the allergen intraperitoneally, they're black 6 mice. There was a fantastic talk from Stephanie Eisenbarth about why this is a problem and why this happens. But if you challenge them intraperitoneally, they'll have an anaphylactic reaction. What we saw when we were looking in this model is that if we gave just a single OVA-CT gavage, that didn't happen. The mice didn't make OVA-IgE, they didn't make OVA-IgG1. But we wanted to know if something changed. We gave them an exposure. Did they just fully ignore this exposure, or did something happen? And so what we thought to do was, well, let's re-expose the mice with OVA alone. If you do that, 3 gavages of OVA, that's a tolerance model. The mice would be tolerant if you did that. So whatever you're seeing is what changed in the beginning, that first gavage. And what we found was that these IgE responses and the IgG1 response were potently boosted by this additional allergen exposure. And then when we take a look at the clinical reactivity of these mice, it was really only the mice that the 4 OVA-CT obviously responded with a drop in core body temperature and an increase in hematocrit, but the 1 OVA-CT mice did not. And when we boosted, you get that, we basically rescued that response. And so what this kind of was saying was that a single insult is sufficient to drive allergic immunity. And you know, we first did that experiment, I want to say probably almost 8 years ago. And it took us a really long time to try and figure some of this stuff out. And in that time, some of that observation had been found by these 2 other papers where Stephanie Eisenbarth's group had found that just giving the sensitization adjuvant in that first exposure was sufficient for the IgE response if they did the OVA re-exposures. And then there was this really interesting observation from Alex Dent's group, and Dr. Dent is here, which is a huge honor to me. I'm actually going to show some of his data. I hope that's okay. He did this really interesting experiment. This was a slightly different model to what we did. They had cholera toxin in both of these exposures, although they did some work where they did peanut alone. And there was a really interesting thing. If you gave this 1 gavage and then you re-exposed with a second gavage 8 days later, specifically 8 days later, you would get an IgE response. But if you did it at 3 or 15 days later, you didn't. And this was interesting to me because now we're starting to get into the territory of the things that I think about, which is memory and how long these things last for. So what I wanted to understand was basically, is this allergic memory that is seated in this like silent phase, is it memory or is it something transient? And I think this is pretty important for studying allergy, actually, because if someone can be exposed to something and we create some transient response, maybe that means that we can shift that response and stop them from becoming allergic if we can identify those people somehow. But if that first gavage is enough to just create lifelong memory that would drive a huge IgE response, that's a serious concern. I'm thinking about myself. If I travel to a place where I've never had the food before, have bad food, I might not know that I've been sensitized, but if I go back again, I might start making a bunch of IgE antibodies and then become anaphylactic. right? So these are really important clinical things that I think need to be understood a little better. So Stephanie, in her talk, said that these allergy models are annoying because they're very long. And I completely agree with her, and I think I'm going to one-up her on that one, because we decided that we were going to try and figure out how long this lasts. So we gave this OVA-CT gavage, and then we waited. And we waited 1 week all the way up to 14 months. And then we took those 3 OVA re-exposures that we were doing, and we just set them back by 1 month, 3 months, 9 months. The idea was to see how long, is this a transient phenomenon, or is this something that is persistent? And what we saw, and this really blew us away, is that when we re-exposed the mice 9 months later to 3 OVA exposures, remembering those would be tolerizing exposures, the mice make a huge IgE response. And when we challenge those mice, they're clinically reactive. And you may be asking yourself, hey, here's the 14-month time point. Why isn't this over here? And the reason is because we didn't anticipate it would last that long, so we didn't do it, and we knew we had to repeat it, so we did a whole another year's worth of experiments after this one. And we don't pool ELISA data because we don't think that that's a good idea, so we have a thing in our supplement that has that 14-month thing in it. So we had to do it all again. And it's a little more variable, but really the mice reacted pretty strongly and they made a lot of IgE. So even 14 months later, but there's no sign. This group right here, this group that is in red, that is one OVA-CT alone re-exposure and a 9-month wait. They never seroconvert. It just stays, which is very, very weird. So I am a card-carrying B-cell biologist. This is what I love. This is what I want to do. And I was trying to study memory in this model. And so what I saw here was a really interesting opportunity. I thought that maybe what was going on here was there's a bifurcation between the plasma cell differentiation, the memory B-cell differentiation, and the rules that decide those paths, those fate decisions, are not very well understood. There's evidence out there to kind of direct these things, but we don't have a comprehensive view. And so what I was thinking was, well, now we have a perfect model. We've made memory B cells, but we haven't made plasma cells, so we can understand the inductive signals. But we kind of needed to show that this was the case. So what we did was we needed to probe the antigen-specific B cells and see what they were. And I was very lucky to meet Justin Taylor at a Keystone meeting in like 2016 or something. And I was talking about this problem that we were having of we were using OVA monomers and we couldn't really see very much. And he said, don't worry, I've got you. I have the perfect system. And he had just started his lab and he was building these tetramers. And so he taught us how to do it, and we've been doing it ever since. And we put together a publication on how to build tetramers the way that we do it. And we think it's a good model because if you take a look at a naive Black 6 mouse, we create these decoy tetramers that have everything except we change out the antigen. So basically, they can be detected on a different flow cytometer detector. And if you look at a naive mouse, when we don't do an enrichment, it's really hard to see what's going on. But if we enrich a whole sample, 100 million cells, we now have enough resolution to look at naive B cells. So we knew basically whatever we wanted to study, we'd be able to see with these tetramers. And what really jumped out at us and freaked us out a little bit was we decided to use these tetramers over the course of this re-exposure. And this in red here is the group where we gave OVA and CT, but we didn't re-expose. And there wasn't an emergence of switched B cells above baseline. And we thought, and what it took was 1, 2, or 3 re-exposures to start seeing switched B cells emerge in this model. And we thought, well, maybe we're just looking at the wrong time point. So we did that one gavage and we looked out and we've gone all the way out to 30 days and it never emerges. We don't see an emergence of switched B cells above baseline. And to show it a few other ways, there's no germinal center B cells in this mice and there's no IgG1 induction either. So you give a gavage, it looks like the B cells don't get activated, maybe. I wasn't comfortable making that claim here, so I designed an experiment where we took a CD40 5.1 mouse, and one of the things we can do with these tetramers is we can enrich B cells out, and we can transfer them. So we stained them with cell trace violet, and the idea was to see if they would divide at all when we gavage the mice. And what we found was when you give OVA alum, which we know drives a huge response, the cell trace violet fully divided, but in the naive and the one OVA CT, there was no division whatsoever that we could see. So the B cells didn't divide. And again, I was still freaked out. So we know after BCR stimulation, they'll upregulate CD86, no CD86 upregulation. So this model, me being the card-carrying B-cell biologist, I wanted to study B-cell memory, but the model was telling me this isn't a B-cell memory problem. So we asked the next question, the question that us B cell biologists never want to ask, which is what's going on with the T cells. And we wanted to know if they were activated. And it turns out that this was a pretty tricky thing to do because the responses are pretty small. So one of the ways we did this, and we did this many ways, there's a lot of data in this that I can't show for time, but we took OT2 cells and we transferred them prior to this gavage just to see if the T cells were activated, the OT2 T cells. And indeed, in this one OVA CT gavage group, we do see antigen experienced or CD44 positive T cells. And so then, there's some great work from Alex Dent and Hirohito Kida and Stephanie about TFH cells being the ones that drive IgE production. So we thought, are these things TFH cells? Did they get that far? And it turns out, if you look at an allergic intragastric model, they don't induce a huge number of TFH cells, but the one OVAC2 group didn't induce any whatsoever. So they didn't become TFH cells, but they are activated. So we just started going down the list. Are they Th2 cells? And we did this a few different ways. We looked at ST2, we looked at GATA3, we even crossed forget OT2 mice to transfer OT2 forget cells, and they never even induce IL-4. They never start producing IL-4. So they don't make ST2 and they make the same amount of GATA3 as the no treatment group. So they don't seem to be Th2 cells either, and they're not transcribing any IL-4. Very, very strange. We've gone down the list. We've looked for FOXP3. We've looked for other things. They don't express any of those markers. And I don't like this OT2 transfer system because it's kind of a gain of function system. We've put a bunch of cells in and we've changed the precursor frequency. So we did this with some MHC tetramers that Mark Jenkins sent us. They're these affinity enhanced tetramers. And we saw the exact same thing with the endogenous T cells, which was, a very interesting observation from my perspective. So this got me to thinking a lot, and we have this memory that's induced. We have T cell activation in the complete absence of B cell activation, which is very odd. And a number of years ago, I was reading a review from Luca Mezzin, Jonathan Ershing, and Gabriel Victoria, where they were talking about bottlenecks in B cell activation. And you have an activated B cell repertoire upon an antigen exposure, and there's potentially a bottleneck prior to the GC where cells will go into the GC, but only certain ones would. Or this was the thought before the confetti experiments that Gabriel and folks did, where it turns out that this bottleneck is actually a pretty weak bottleneck. Many cells of a variety of affinity get into the germinal center. And then we start to select these cells to create higher and higher affinity germinal centers. And what I think we've kind of stumbled onto here, and one of the reasons I think this matters, is because it seems like there's actually a bottleneck number 0 here that maybe we weren't super aware of before, which is that in some exposures, the immune system may choose not to activate the B cell response. And that kind of makes sense to me because germinal centers are very cost intensive, they're antigen intensive, and sometimes maybe we don't want to go all the way. We want to stop and say, well, we'll make some T cells, and then we'll just kind of wait. And if we see the thing again, we can drive a bigger response next time. That notion that I'm kind of proposing here and stealing Gabriel's beautiful visuals relies on the notion that CD4 T cells are actually required for this. So we decided to test this. And I've already been pushed out of the B cell space in this story. I was really nervous that I'd be pushed into trained innate immunity and I would even be studying adaptive immune cells anymore or something. So we did a pretty simple experiment where we depleted CD4 T cells either around this priming exposure or around the re-exposure. And what we ultimately see is either of these depletions completely ablate the IgE and IgG1 response. So we know that CD4 T cells are required at both of these stages. And I think another thing that's interesting is if we had seeded some IgG1 memory that we just hadn't caught with our tetramers, I would anticipate that those cells probably would have been allowed to divide and make more IgG1 in the re-exposures, even without T cells. So we really don't see any IgG1 here, which gives me more confidence that this T cell repertoire is just not activated at all. Or sorry, the B cell repertoire is not activated at all. Anyway, when we challenge these mice, we see the exact same phenotype that only if we deplete T cells at either time, we completely lose the phenotype. So this is one of my favorite things to do in a talk. We're going to do a 7th inning stretch summary where I'm going to kind of talk to you about where we're at so far so that I can cool down too a little bit. We have this first gavage that we give a mouse, and we activate, it seems, a memory T cell population but we can't figure out what they are based on their phenotyping. And we induce we don't induce any B cell activation. And so it seems like in these OVA alone re-exposures, which would otherwise be tolerizing if we hadn't done that first gavage, that the way that this model may work is that the T cells are actually driving B cell activation during the recall. So all of the programming that those T cells would need to create a secondary IgE response already exists within the memory T cell repertoire. And so those cells would then go on to create IgE plasma cells, let's say. So we wanted to test this very specifically. And the way that we chose to do this was taking advantage of some very old linked recognition work where you can sensitize with one antigen and then re-expose with a linked antigen to see what happens. And so what we did was we gave this OVA and CT gavage, which we know will create these memory T cells that are OVA specific. But when we came back with our re-exposures, we gave NP conjugated to OVA. And the idea is that these mice have never seen NP before. So that's a completely naive NP specific repertoire. And when they capture the NP, they'll present the antigen to the T cells and we'll be able to see what happens. And basically, if these T cells can hold IgE memory, they should drive an NP specific IgE response. And basically, that's exactly what happened in this system. So our positive control was NP-OVA in both of these gavages. And this in the orange here is an OVA-CT first gavage and then a re-exposure with NP-OVA. And then they make IgE and they make IgG1. But when we swap out the carrier and now we use NP-BSA in the re-exposure and the T cells can't talk to the B cells, we don't get the IgE or IgG1 response. And if we give NP-OVA alone in the re-exposures, because we were worried that that might be sensitizing in some way, the mice don't become sensitized. So when we challenged these mice, we challenged them with NP-BSA. And the idea there was because we didn't want the OVA reactivity to change the system. So basically, it's only the reaction to NP. And what we found was that the mice only that received NP-OVA during the re-exposure, not NP-BSA, were clinically reactive upon challenge. And this kind of is telling us that these CD4 T cells can hold the humoral memory, so that themselves, we don't need memory B cells in this system to hold an IgE response. Which I think is a pretty incredible observation, and also frustrating observation because I like memory B cells. So that's the other pain. So, we've been trying to dive in a little bit more into what's happening here. And there was a paper that came out from the Jenkins group where they were studying gliadin-specific T cells. And what they had found was that when they did a tolerance model where they fed gliadin and they looked at the T cells, they created what was a TH, they called a TH lineage negative population. So something that looked pretty ambiguous like what we were seeing. But I very specifically haven't called this that for a reason that I'll tell you right now, which is that in this model, when Jenkins comes back and tries, first he's tolerized and then comes back and tries to create the immune response in these mice, the Th1 response doesn't happen. There's some defect in there that stops that Th1 response. And these cells are shunted into like a Treg fate pretty much. But in this circumstance that we have a similar looking cell, but it can drive an IgE response, virtually the exact opposite phenotype or the exact opposite function. And this was a really tricky thing for us to untangle. So what we wanted to do, and I'm realizing I didn't change the title of this slide, but this is not that. We're trying to understand what the TFH cells or what the T cells are doing. We had to create this system where we were able to actually get enough cells to do some transfer experiments because these responses were really, really small. So we had Emily Grudjushko gets all the credit for this because these were hard experiments and she was a tank through all of them. But she basically would do a first transfer with OT2 cells and create these prime T cells, and then would go and fish back out those prime T cells and transfer them again into a new host. And we had tried doing this a number of different ways, but the model system that we wanted was that we could gavage the mice with OVA and they'd have a response. But the problem is that there's no adjuvant in this system. It's a very small push for these cells, so they don't end up dividing enough to kind of fully recreate the immune response. So instead what we did was we put them into TCR beta knockouts, and then we drove the cells really hard. We gave them an IP with OVA and alum because we just wanted to see what those T cells could do. What was their programming? And this ended up being really insightful because what we found was that when we gave naive OT2 cells in this transfer, and then we IP immunized, we had a TFH response. But when we gave primed non-TFH cells, we were very careful. We sorted out anything that looked like it could be a TFH, so anything that had really any signal for CXCR5. And we transferred when we transferred these prime cells into the host and we IP'd them with alum, there was a huge induction of CXCR5 positive T cells. So these things had an intrinsic program that drove them to become TFHs very, very potently. And this is seen here where up to 60% of the activated T cells were TFHs in this context. And well, they were CXCR5 positive, maybe like pre-GC TFHs by comparison to the TFH cells where the signal wasn't quite as clear. So we've done something here where we create this T cell that is pretty ambiguous. It's never seen IL-4, it's never made IL-4 before. It's never been a TFH before, but it knows that it's supposed to become a TFH and then drive an IgE response, which is pretty incredible. And so I have this slide for if I have time, because we have a lot of mechanistic experimentation on this with different blockades and stuff. But the one that kind of baffled me the most was we just wanted to double check if this was an IL-4 receptor dependent process. And so what we did was during this OVA-CT gavage, we blocked IL-4 receptor alpha with an antibody. So the T cells should not be able to see IL-4 signaling at that time. And that didn't have an effect on creating IgE, IgG1, or hypothermia. So the induction of these cells does not require signaling. It's known that IL-4 is not needed to make a Th2 cell. You can, there's discussions around this, but basically a dendritic cell can talk to a T cell and seem to induce what looks like a Th2 cell, even if IL-4 is not present in the system. But what I don't think I can really wrap my head around is how this cell has never seen IL-4. It can't have seen IL-4, but it knows that it's supposed to make IL-4 in the future and drive an IgE response. So there's something pretty fundamental about this memory that we don't fully understand just yet. And the programming of these cells is such that they are capable of holding TFH memory despite never having been that cell before. It's a very interesting phenotype. So I'm going to stop here and take questions, but the conclusions of this are a little concerning for me as a human being, because it seems like one bad exposure is all it really takes to knock you down for life with allergy. You could just have latent allergy in this context. And I think this is going to be enormously difficult for us to figure out in humans if it's even possible, but it could be that. It's just one exposure. And another big thing for us is that it seems that memory CD4 T cells hold IgE memory through de novo activation of naive B cells. And you don't need a memory B cell in that context to do this function. And we have another paper that was just published recently where we made some clonal connections between IgE and the memory B cell compartment. So we know that they can contribute, but the vast majority of IgE sequences don't make any clonal connection. So it's possible that really the CD4 T cells do most of the IgE memory. The programming instilled in these cells happens prior to TFH differentiation or IL-4 production, which is very strange. And a big paradigm is that there's many layers of immune memory that we need to worry about when trying to interrupt these different immunopathologies. Even in autoimmunity, we have to inactivate many layers of memory. And so that's everything that I have. Thank you again to BioLegend. Emily Gajrushko was huge for this. My PhD mentor, Manel Jordana, was also I did a lot of this work when I was at various stages. We would get stuck and then wait a year and then come back. And we had to wait for mice for 2 whole years twice. So, there's been a lot of stages to this project. But I think we're finally at a point where we understand what's going on with it. Thanks very much. Questions from people? Yeah.
In your adaptive transfer experiment, in which you took the naive TFHs you put in the other mice to challenge, were the naive TFHs enough to induce? I mean, if you checked, were they enough to induce the CD23 L4 RA high B cells, memory B cells?
So we haven't really looked for that so much in this model. We've been more focused on trying to understand. I've kind of given up on the B cells after they were completely naive after the exposure, if I'm being honest. But from any of the other work we've done, anytime we see IgE present in the system, we always see these IL-4 receptor very high, CD23 very high. We call them MBC2s, which was the paper I was talking about that was published earlier this year. They're always present in these various systems, and the only time that I have seen a complete absence of those cells in the antigen-specific repertoire is when there's no IgE induced when you immunize, and that's the context of CpG immunization. It's the only system I know of that just makes zero IgE whatsoever. And I think there was a poster about that here, actually, which is kind of cool. More questions?
Very interesting, Josh. So I was thinking about these T cells with memory that you say that can produce IL-4. Do you know how polyclonal are these cells, and what are your thoughts? Have you looked at the epigenetics on that, and maybe a toxic or something?
Yeah, that's a fantastic question. So, you're basically asking me what are these things. In terms of polyclonality, a lot of the work we've done is in the OT2 system, so we can't really see into that. We've used the tetramers a number of times to look at these cells. It's a pretty small response on the order of a few hundred cells, so it may be quite polyclonal, or it may just be one clone that chose to expand. It's a very good question, actually. It's something we should look at. We're very actively, and we have some data on this, but we're very actively working on getting a transcriptomic paired multi-omic profile of these cells, which we've done the math. We have to do 70 mice to get the experiment. So my whole lab and 2 other labs are signing on to help to do that. So it's going to be fun, but I think it's going to be very insightful. My expectation is that the way that this memory is held is epigenetically. So they've been instructed to basically loosen these repertoire, or the genomic loci at these different IL-4 sensitive sites, and then they can open them very quickly when they're re-exposed.
You kind of answered my question, because I was going to ask about the transcriptome and just trying to find out more information about them. But I have a separate question. First, fabulous work, very, very interesting. I wanted to know if it is possible. This is a question. I'm more a T cell guy. I'm not a B cell guy. Is it possible to do co-culture? Because you tried many ways to make those B cell respond, but now if you put those T cells with them ex vivo, is it possible to have this response ex vivo?
Good question. So we actually have some ex vivo work that's in this, where when we culture splenocytes with an allergen, for example, you'll see IL-4 and IL-5 production, for example. That work was done many years ago by a postdoc who predates me in the Jordana lab, so I don't think we looked at IgE, which is maybe something we should have done. But the responses we get out are very small. That's the thing. I mean, to get one well with enough cells in it, we may be doing lots of stuff, right? Or we're pulling them down by their tetramer specificity, which on T cells is a little bit of a no-no. So let's talk later, because maybe you can help me figure that one out. Yeah, Mark.
Very nice presentation and amazing insights into how immune system works. I like the final conclusion. The first bullet, it's all one bad exposure to allergen. That's all it might take. Could you define what that bad allergen? Is there a good allergen exposure? That's all it might take for us to be all right with that allergen. That's number one. Number two, the problem with the system. You have a system where it's not just the allergen you are putting in. You have a cholera toxin. So how do you put that into the equation of all the things that you have seen? It could be just simply an artifact of a cholera toxin coming into the system.
Yeah, that's an amazing question, the second one, and one that we were very, very worried about. We've used many adjuvants. So if you do the same thing with CpG, you won't make an IgE response, but you'll have the exact same priming phenomenology, where you will only get T cell activation, and when you re-expose, it'll drive an IgG2b and 2c response. So I think maybe more than adjuvant specific, it may be route specific, because one way that we have been able to overcome this is if we gavage mice with 100 milligrams of OBA and 5 micrograms of cholera toxin, then they'll make IgE after that first gavage. And there's probably some connection to what Stephanie Eisenbarth was talking about, which she said not to post slides, so I'm not going to say any more than that. But you can talk to her about some of the stuff that she's been working on. She, you know, it may be that there's some intricacies to how the gut works that can allow for such a disparate connection between the B cell and the T cell response, which I think is very interesting. Related to the first point of, is there good exposures? Yes, I think there are. If you look at the LEAP trial, if you feed people peanut when they're very, very young, there's like a 13-fold decrease in the rate of allergic sensitization. So it seems like maybe the biggest problem in this system, or in allergy in our biological system, is if we get that exposure, that's a bad exposure. And who knows what that could be, right? I mean, it could very well be, I mean, people don't have cholera, so it's not cholera toxin itself, but we have many similar bugs that create similar toxins. So it could be that. Could be many adjuvants. Yeah, right. Yeah, the concept there is that in India there is still cholera, but there isn't sensitization to foods. I suppose as far as we've checked, what time's this end at? 3 or 4? 3? Yeah. Okay. Is there more questions?
Well, yeah, that was really very interesting. And obviously, we've done some very similar work. So I had several questions, and maybe it's too much for right now. But when you looked at the Th2-ness of the T cells, were you looking at total T cells? Because I mean, obviously, TF8 cells make a lot of IL-4, and they seem to develop the IL-4 when they turn into TF8 cells. So it wasn't clear if you were looking specifically at TF8 cells there. But
We were looking within the OT2 cells as a whole. So they were OT2 foregut cells, and when we transferred them, and we looked at all the OT2 foregut cells, and none of them made IL-4 after that first gavage.
I mean, when we've done OT2 transfers in a similar study, and actually not that many total of the transferred cells turn into TF8 cells, so that might be part of what's going on. Have you looked at Tregs or in this model and what they're making? Because you can get IL-4. Oh. Okay. Okay. Well, one last thing is, you showed our model is a little bit different because you did a month after the priming, but shoot, I forgot what I was going to say. No, the antigen swap out with the hapten. So you did the NP hapten. We do a week after, whereas you do a month. And like if you go a week after that, it doesn't matter which antigen the T cells are responding to. So whereas you saw a very precise need for the same antigen specificity. So it's different, but it's different systems too.
The more that I've kind of looked at both of our work, the more I think that they're not the same system. Because you very clearly show in your work that you have activated B cells. Like you have Verigem expression after that first gavage and germinal center cells that are ongoing. We don't see any of that. So I presume that the antigen won't matter as much if they're stimulating those cells through their B cell receptor, right? But B cells are naive. If you stimulate them through their B cell receptor, they would tolerize, they would clonally delete, or they would ignore, right? So, you know, I think I really like the work that you've done, by the way. I'm a huge fan of it, and I go back to it very frequently. But I think that the circumstance of having this completely unactivated B cell repertoire makes this, as a thought experiment and as an immunological experiment, very insightful. That we can completely separate those two responses and deal with them separately, and that may be spatial, that may be macroanatomical, that may even be they are engaging and just choosing not to activate. I don't know at what level that happens yet. Sorry, there's a question back
here. Great talk. Kind of going along with the previous question about the adjuvants and I'm familiar with cholera toxin, not as much the other adjuvants and their mechanisms, but have you considered or have you blocked epithelial alarmins like IL-25? Probably won't be directly acting on the T cells, but to give some insight of other signals that could be priming this response.
So I think to answer your question, I think the limitation of the work, or maybe I wouldn't say limitation, I would say future direction of the work, is we haven't done a lot of work trying to understand the inductive features of this T cell phenomenology. We've been more so focused on the longevity and the memory, primarily because that's my lab, right? I think it's enormously interesting, though. And those would be the types of experiments that I would start with. I think blocking alarmins would be a really great start. There's some folks here that study tough cells, and I think there's probably some really smart interactions with that might make a lot of sense in the system. Any other questions? Well, if not, thanks so much for coming. I really appreciate the attendance.